Good morning or good afternoon. Welcome to good evening, and thank you for your participation. Before we begin, I'd like to say something about ISSUP Italy. My name is Giovanna Garofalo. Before we begin, I'd like to read an introductory note wanted by the ISSUP webinar disclaimer policy. Please note that this webinar is provided for informational purposes only. It does not constitute medical treatment or treatment advice. I punti di vista presentati, the views presented here are those of the speakers and not necessarily those of ISSUP. Please allow me a few words of thanks that I would like to make, not out of institutional duty, but out of sincere gratitude. First of all, I'd like to thank the ISSUP global team, Jeff, Livia, Kirsty, Olivia, John, and the entire technical group that enables the national chapters worldwide to present topics and share experiences of great interest in the field of prevention and treatment of substance use disorders and related topics. Also, thanks to the speakers today, Professor Carla Cannizzaro, full professor of pharmacology and director of the university, delegate of the rector of the University of Palermo for academic policy for prevention and treatment of pathological dependencies. She's an expert consultant on the Italian National Institute of Health. So welcome, Carla, and Dr. Anna Brancato, the assistant professor and manager of the neuropsychopharmacology lab of the University of Palermo, expert component of a bioethical committee. They have been producing a lot of papers. They are authors together on more than 160 scientific works published on high-impact peer-reviewed journals. Professor Cannizzaro and Dr. Brancato have been carrying out research activities in the field of substances of abuse for many years, and I thank them for their prompt availability to present today's topic for Casa Rosetta and for ISSUP Italy. This meeting is part of an ongoing collaboration in the field of education aimed at the community of professionals at national and international level, with the planning of masters and advanced training courses. Thank you to both of them. I would like to extend a special thanks to the president of the Casa Rosetta Association, Giorgio De Cristoforo, has made available the pathological addictions operative team of Casa Rosetta and has enthusiastically accepted the commitment to host the ISSUP Italy, putting the association's resources and experience in the field of addictions at the disposal of the international network. Of course, I'd like to thank again, reiterate my thanks to the president of Casa Rosetta Association, as I said before, Giorgio De Cristoforo, because this has been possible thanks to him. The national chapter Italy is involved in the international network with the resources and experiences of the association in the field of addictions. The Casa Rosetta Association is a non-profit organization based in Sicily that has been working in the field of pathological addictions for over 30 years, with counseling centers and therapeutic communities in Italy and Brazil, following hundreds of people with substance use disorders and their families, as well as training and awareness activities carried out in various countries around the world. In particular, the training and awareness campaign carried out in Tanzania in recent, thanks to the funding from UNODC and the US State Department, with the valuable collaboration of the Tanzanian government and the Muhimbili University in Dar es Salaam. My heartfelt thanks go to Pietro Cipolla, a member of the board of directors of Casa Rosetta, who is passionate about and supports us in all the international activities that the association carries out in different countries. Thank you, Peter. Together with Peter, I thank the colleagues of Casa Rosetta and FIGT who are part of the communication staff and who animate the national chapter ISSUP Italy. And last but not least, and today is the right time to say thank you to our expert translator, Mr. Carmelo Donato, who has always accompanied us in our international activities, guaranteeing international linguistic support with professionalism, passion, and I may say affection for our association. Thank you, Carmelo. A short presentation of today's webinar. The title for this webinar is Early Exposure to Cannabis and Long-Lasting Consequences in the Brain. Professor Cannizzaro and Professor Brancato will show evidence on the consequences of exposure to cannabinoids during development and prison data, supporting possible intervention strategies. The acceptance of the use of cannabis and cannabinoids in the population, both for recreational purposes and for self-medication, is growing worldwide, with a consequent increase in risks among the most vulnerable subjects, such as adolescents and pregnant women. The speakers will talk about how early exposure to drugs of abuse and cannabinoids in particular interferes with key neurodevelopmental processes, producing lasting consequences in the brain. And now, with great pleasure, the floor goes to Professor Cannizzaro, who introduces us to the topic on the use of cannabis during vulnerable times. With question and answer, you can submit your questions with a question and answer function or box. In the message box, you can find a link for listening to the interpretation from English into Italian. Thank you. Professor Cannizzaro, you have the floor.
For this invitation, we are very happy to be part of this important event, and I'm going to start my presentation. And yes, here we are. Okay. Just let me start by a quick so just by a quick glossary in order to give you a better way of understanding the subject that we are treating together today. And so we are going to talk about cannabinoids, every chemical substance that is able to bind to the cannabinoid receptors and have similar effects to those produced by the cannabis sativa plant. We will be talking about neurotransmitters, the chemical used for neuron communication, such as dopamine, GABA, and glutamate. And neurotransmitters bind to the receptors in order to get their activity. We'll be looking at the synapse, which is the site of neurons communication, and we will see the synapse is composed by a pre and a postsynaptic site and a cleft between these two sides of the environment. And especially we'll be focusing on some specific areas of the brain where cannabinoids and cannabis are able to exert their important relevant activity, such as the mesolimbic system, which is the brain system where dopamine is released in order to produce a reward and the feeling of pleasure. The PFC, the prefrontal cortex, a region of the brain so crucial, so central, the site of executive functions and inhibitory control, the hippocampus, the site for learning and memory processing, and also the HPA axis, the stress axis, which is just responsible for the activation of the stress response as a consequence of the exposure to stressors of different nature. So we obviously talk about cannabis, which is the most popular illicit drug worldwide, although illicit is not a proper term anymore since the widespread legalization of cannabis has interested many countries in the world. Periods of lockdown during the COVID-19 pandemic drove increases in the use of cannabis in terms of both amounts used and frequency of use. But it is also important to recognize that cannabis legalization in North America has increased daily cannabis use, especially potent cannabis products, particularly among young adults. And actually, young people do like cannabis the most among other substances of abuse, as this important study, the European School Survey Project on Alcohol and Other Drugs, has shown. This is a cross-sectional study which has interested students aged 15 and 16 years and is about drug use and other forms of risky behaviors, and is carried out every 4 years in Europe. And actually, the ISSUP students, when questioned about the starting of their cannabis use, 2.4% of these students reported the first use of cannabis at very early stages, such as a 13-year, and 35% among the students who reported cannabis use in the last year are considered to be at risk of developing cannabis-related problems. Actually, cannabis consumption among the youngsters probably is promoted by the so-called influencers, celebrities, who just like to share their feelings about cannabis on the web. And they talk about the feelings of pleasure. They talk about the stop thinking activity of cannabis, the stress relieving property of cannabis. They experience when they smoke cannabis several times per day, a substantial number of joints for a long time. And we could also ask ourselves, why does cannabis make users feel good? Well, the reason is that cannabis is able to stimulate a particular brain region, which is the reward mesolimbic pathway, where tetrahydrocannabinol, which is the main psychoactive compound in cannabis, is able to induce dopamine release. And dopamine release from the ventrotegmental area of the mesolimbic pathway is very relevant, because when dopamine is released, a feeling of pleasure is perceived, and this reinforcement is the very basis for the repeated consumption of cannabis, but not just cannabis, also the other substances of abuse, because cannabis actually shares this activity in releasing dopamine in the mesolimbic system from the ventral tegmental area into the nucleus accumbens, share this effect with many other substances of abuse, with different mechanism of actions, but the release of dopamine in the nucleus accumbens is the key to understand the reinforcement. The chronic and the repeated use of cannabis, on the other way, is able to produce significant morphological and functional alterations in the brain circuitries, which can disrupt reward sensitivity and motivation, and this is thought to represent the basis for cannabis dependence. And this is something similar to what we have described for other substances of abuse, such as morphine and ethanol during withdrawal, during abstinence. So, cannabis sativa is the main source of the phytocannabinoids. So in the cannabis sativa, we have lots of substances, because besides the 100 of cannabinoids present in the plant, we have also other kind of substances such as the flavonoids, terpenes, fatty acids, and all of them contribute to the so-called entourage effect of cannabis. But the most studied and the most characterized principles, active principles in cannabis are definitely tetrahydrocannabinol, that we will call THC, and also cannabidiol, cannabidiol CBD. They have a range of opposing effects on the human brain and also on behavior. Indeed, if THC is able to acutely produce a euphoria, is able to impair learning and produces psychosis-like effects and increases anxiety, on the other hand, CBD can enhance learning, exerts antipsychotic and anti-anxiety properties in humans. So, when taken together, CBD can ameliorate the harmful effect of THC, but this buffering capacity of CBD is strictly dependent on the ratio between THC and CBD in cannabis. Unfortunately, in the last 2 decades, the percentage of THC in cannabis samples has increased dramatically, and it ranges from 15 to 30% of the content. So, on the other side, the CBD is moreover the same. Therefore, it means that THC and CBD ratio has increased 80-fold, increasing the risk for harm. In order to understand how the phytocannabinoids, THC and cannabidiol, are able to impact on the many and important relevant functions, not only in the brain, but also in the organism, we have to talk about the similarity between the phytocannabinoids and with endogenous compounds that we call endocannabinoids, basically anandamide and 2-arachidonyl glycerol. It is called also 2-AG, easily. So these endogenous cannabinoids are able to exert many functions in the brain, but also in the organism. The endocannabinoid system is one of the main regulatory systems that we have. The endocannabinoid, anandamide, and 2-AG are produced in the synapse, are produced by neurons after they are excited. So they are produced, they are retrograde lipidic mediators that cross the synaptic cleft, which is the space between the neurons, and bind to the CB1 receptors, cannabinoid 1 receptors, which are mainly expressed in the brain. Endocannabinoids play a crucial role because they are involved in the suppression of synaptic transmission, which means inhibitory and excitatory transmission through multiple mechanisms, independent of the synaptic type or transmission duration. And they do that when it is necessary. They are called on-demand neurotransmitters because they are released when the neurons need it in order to provoke the suppression of the inhibitory or the excitatory transmission when it is needed. So let's go and see where the CB1 receptors are expressed. They are not only in the brain, but also in the liver, not just the CB1, but also the CB2 receptors. They are in the liver, they are present in the reproductive system, in the cardiovascular system, in the skeletal muscles, in the gastrointestinal tract, and they modulate many functions and also nociception and immune modulation. So the activity of the endocannabinoids, the activity of the endocannabinoid system is very wide, and it doesn't affect the brain only. But if we want to look at the CB1 receptor expression in the brain more deeply, we see that we have a wide expression in the cerebral cortex, where the endocannabinoid can modulate cognition functions such as decision making and the emotional behavior. They are expressed in the basal ganglia, the caudate nucleus, the putamen, the globus pallidus, where the endocannabinoids are able to modulate movement, but also various types of learning. They are expressed in the amygdala, which is the region where the feeling, the fear, the stress perception is processed. So cannabinoids can really modulate amygdala functioning, and also in the hypothalamus, modulating and regulating feeding and the neuroendocrine function. They regulate the hippocampal memory processing, and also the cerebellum related motor control, and also dorsal vagal complex being able to regulate emesis as well. So this is quite helpful in understanding, in letting us understand the psychotropic bioactivity of THC in humans, because the THC bioactivity ranges from the somatic to the perceptual and the cognitive domain. So if we want to understand the effects, the wide range effects of THC, we have to remember that THC binds to CB1 receptors, and CB1 receptors are expressed everywhere and in relevant regions of the brain. Psychosocial stress is the main stress that the young people, the adolescents, have to face. And psychosocial stress coping is really demanding, is really exhausting, and sometimes psychological stress is able to produce anxiety. So anxiety also is the result of the activation, of the abnormal activation of the HP axis, which is the structure in our brain, in our body, which is able to activate, to produce an adaptive defensive response to stress. So when we receive stressors, our HPA axis, hypothalamic pituitary adrenal axis, is able to release the so-called stress mediators such as glucocorticoids or CRH, and these glucocorticoids on their hand are able to induce the fast increases in the endocannabinoid synthesis in brain areas that shape the perception of stress, such as the prefrontal cortex, the hippocampus, and amygdala. Why glucocorticoids enable endocannabinoid release? Because endocannabinoids are able to counteract excessive HPA axis activation, and thus they limit the release of the stress mediators such as CRH from the hypothalamus and so on. So this is one of the main reasons for cannabis consumption being anxiety relieving. When the stress axis is activated, the endogenous cannabinoids can be produced in order to tune down the stress response. Now, I wanted to show you one of the first, maybe this is the first study about self-medication with cannabis among adults, young adults, with legal access to cannabis after cannabis, recreative cannabis legalization in Canada. And this study shows that the young people use to self-medicate with cannabis the most, and they use cannabis for different conditions such as psychological disorders, somatic disorders, and anxiety is one of the main conditions they self-administer cannabis for, just to keep calm. And when we talk about young people, we talk about not just not yet adults, but we have to understand that the brain changes, and the brain changes not just in size, but also in the way it works. The brain changes morphologically and functionally across all the ages. And it is important for us to remember that the human brain still matures after adolescence, because sometimes we think that adolescence is the period in between 10 and 19 years, but this long maturing process for the brain lasts beyond. So we will have a full mature brain after 25 years. So if you look at the brain maturation, we will have an early maturation of the limbic areas, which are the hot areas of the brain, sensitive to reward, sensitive to pleasure, sensitive to sensation, and a delayed maturation of the so-called cold brain areas, such as the prefrontal cortex, which exerts a top-down control over risky behaviors, impulsive behaviors, so that we arrive at the proper maturation after 20 years. And it is important to remember that the connection between these different parts, to different parts of the brain is very important, must be maintained, must be protected. But we also know that the exposure to multiple drugs of abuse, including cannabis, during this vulnerable epoch of development may alter the trajectory and may in lasting consequences on brain structure, function, and also in the expression of behavior. Why can we correlate the brain development to the cannabinoid exposure? Well, the thing is that the endocannabinoid system changes dynamically across neurodevelopment too. So we have different expressions in the receptors. CB1 receptors, for instance, progressively decrease from early gestation to birth and reach maximal density, reach maximal densities during adolescence in various brain regions before decreasing to sustained high levels in the adult brain. And the neurodevelopmental events which occur from gestation to early adulthood are regulated by the endocannabinoid signaling. And during gestation, we see neural generation. We have migration followed in a later stage by synaptogenesis and synaptic transmission that continues into infancy, childhood, and adulthood. In adolescence, we have more the synaptic pruning phenomenon that shapes the mature brain architecture with dynamic fluctuations of component of the endocannabinoid system. I want to stress that the endocannabinoid system is therefore relevant for different and precise processes during neurodevelopment, such as neurogenesis, neural migration, synaptogenesis, the fine-tuning of neural circuitries, and the maturation of prefrontal cortex, which means the correct connections between the areas. So the endocannabinoids play a fundamental role in neurodevelopment because they interfere, they can promote all these different phases of neurodevelopment. If we have the exposure to cannabis at different growth stages, from pregnancy to young adulthood, we may affect neurodevelopmental processing, and in this way, we can exert a long-lasting impact on physiology and behavior, which of course can result in diseases, disorders, and abnormal behavior. So I hope this is quite clear, and it is very important to remember that the endocannabinoid system is very much relevant for neurodevelopment. Now I will try to discuss the last slides about the acute and the chronic effect of cannabis, because we have to distinguish between acute rewarding effects and chronic effects of cannabis. The acute rewarding effects are played by an increase in dopamine release and an attenuation of GABA and glutamate release. This is why we have this feeling of pleasure, which is the substantial basis of repeated cannabis consumption. But at the same time, acute adolescent cannabis is able to disrupt cognitive functioning, operating on other neurotransmittorial systems such as acetylcholine or noradrenaline. As time goes by, we have noticed a decrease, an important relevant decrease in CB1 receptor availability in the brain of chronic cannabis users. And this reduced availability regards relevant brain regions, such as the temporal lobe, the cingulate cortex, where decision-making, executive function, the nucleus accumbens, where reward sensitivity is played. And this reduction goes to the cannabis use frequency. So we have a greater reduction in heavy users and moderate users rather than in low users. On the other hand, chronic THC exposure is also able to disrupt the mesolimbic system, but by reducing dopamine availability. So if acute THC is able to increase dopamine, chronic THC is able to reduce dopamine. This reduced dopamine availability together with other modifications in the endorphin, in other actors of neurotransmission or the stress response, such as CRH in relevant regions such as amygdala, these deficits in the mesostriatal dopamine function may contribute to the negative affective state, which is associated with THC abstinence. So different modifications, not only dopamine, but also the opioid system and the stress system can result in negative affective state associated with the THC abstinence. But also chronic THC exposure can result in a persistent disruption of hippocampal and cortical signaling. And in this way, chronic THC is able to affect memory formation and executive function. THC is able to alter the inhibitory excitatory balance in the brain, and this is a detrimental activity for memory formation and executive functions. At last, this is my last contribution to the talk. I want to just remind you that actually the genetic expression, the expression of genes, which is something very delicate, it occurs in DNA, it occurs in the nuclei of the cells, is sensitive to epigenetic, which means that the expression of genes can be altered by relevant environmental stressors, relevant environmental stimuli. So in particular stages of development, such as during perinatal and adolescent neurodevelopment, the chromatin structure, so DNA basically, the chromatin structure is in an open state. So I want you to visualize that the open state means that the chromatin is open to epigenetic and transcriptional machinery. And this state, this open state, is also vulnerable to developmental cannabis exposure. When the developmental process go on, such as in adulthood, the chromatin structure is different, is not open anymore, and is less sensitive to epigenetic impact. That's why the adult brain is less sensitive to some insults than the adolescent brain or the perinatal brain. And there is another important thing that I want to stress. The germ cells are subjected to reprogramming, and so they are subjected to epigenetic and transcriptional machinery as well. And in this state, they are very sensitive to cannabis insults as well. And when they are reprogrammed in an abnormal way, we can have intergenerational transmission from the mother to the child. And this is something we cannot really explain from a molecular point of view, but it is really able to alter the physiological and the behavioral development of the offspring. So with this last slide, I end my contribution, and I am very pleased to leave you to Dr. Anna Brancato, who will go deeper and deeper in the modification produced by cannabis in the perinatal and in the adolescent time window. Thank you.
Thank you very much, Professor Cannizzaro. I'm happy to contribute. Would like to share my presentation as well. Yes, okay, sorry, just trying to making this work. Here we come. Okay, believe you can see my screen. So yes, I would like to expand upon the brain-based risks for cannabis use during adolescence, and this to include these acronyms actually has been suggested, the DAMB acronyms. This stands for dependence, driving, underachievement, mental illness, and bad to worse. So we start with the first word, dependence. Although cannabis is believed to be non-addictive, actually a large percentage of global substance use admissions are related to cannabis. The long half-life of THC contributes to this misperception. And indeed, a long time interval is required to mark to sign the occurrence of cannabis, of marijuana-induced withdrawal syndromes. This is actually well characterized, and the decrease in CB1 receptor availability in mesolimbic, mesocorticolimbic brain regions is responsible for the symptoms which are included in the cannabis withdrawal symptoms, with cannabis craving, irritability, increased aggression, but also depressed mood and anorexia. And a lot of these symptoms are delayed, so they are not perceived as correlated with cannabis withdrawal, but these symptoms are long-lasting, and they clearly contribute to the ongoing use, making the efforts for cessation aversive. So the second word for the dump consequences is driving. Indeed, cannabis smoking can jeopardize the skills necessary to driving, and the fMRI, the functional magnetic resonance investigation in the brain, can detect the lower blood oxygen level responses after cannabis smoking in many brain regions key to the executive control, salience attribution, which are actually impaired. This is an underestimated, undetected phenomenon, and it is very important actually to test drivers not only for alcohol but also for cannabis and other substances of abuse. We did that under the auspices of the presidency of the Council of Ministers for the project Drugs on Street. Indeed, during a weekend night, we tested the drivers suspected of psychophysical alterations for the alcohol recent use, but also for drugs by testing their saliva. Well, the young drivers that were actually tested were positive for like 48, more than 48% for at least one drug, and 94% 0.9% tested positive for THC, so a very high percentage. But the risks for early cannabis use include also its interference for the optimal development of the adolescent brain. And indeed, underachievement is one of the most well-supported correlate of persistent or regular marijuana use during adolescence. A growing number of studies demonstrates that cannabis, the marijuana use, is associated with a decline in academic achievement, in academic motivation. In addition, this suggests a decline in effort-related learning that has been highlighted also in twin studies. This may not surprise you, as we already have heard from Professor Cannizzaro. Chronic marijuana, chronic THC exposure changes CB1 receptor density in the brain, in brain structure actually involved in cognitive function and motivation. Thus, the altered balance of inhibitory and excitatory neurotransmissions in the prefrontal cortex, also decreased acetylcholine release in the prefrontal cortex and hippocampus, are responsible for the deficit in working memory, attention, planning, decision making, so executive function, but also in learning and memory. Plus the decreased dopamine inputs from the ventral tegmental area to the nucleus accumbens, the brain regions involved in motivation, in salience attribution, is responsible for decreased effort to obtain the results. It is important to point out that the age of the onset, the early initiation of cannabis use, is a key determinant of the consequences of cannabis use during adolescence. Indeed, it seems that the early onset, the age of onset, is important for significant deficit in many cognitive functions, in associative learning, memory retention, and academic performance. This is directly linked to the impairment, to the detrimental role that chronic cannabis can have during the early stages of development, because of actually the role of the endocannabinoid system. From the perspective of harm reduction, a major goal would be to delay the age of the first exposure to cannabis. So a few years ago, Professor Cannizzaro led an important project dedicated to the training of middle school teachers, which were trained on drugs of abuse and addiction. We believe that the empowerment of the educational system can be a key to the prevention and the contrasting of early cannabis use, also early use of other drugs of abuse. But we should point out also that cannabis is a known modifiable risk factor for the schizophrenia, for psychosis. The imbalance in the excitatory inhibitory tone in relevant brain regions, such as the mesocorticolimbic brain regions, is related to early and heavy THC exposure, and early, so adolescent, and heavy THC concentrations can actually promote the occurrence of psychosis otherwise latent in at-risk population. We should note that the recent increase in THC concentration and with lower cannabidiol presence, concentrations in the cannabis preparation, in the cannabis plants, can play a relevant risk in this kind of mental illness promoting effect of cannabis. And I would like to show you a recent cohort study from a Danish population, according to which approximately 25 up to 30% of schizophrenia cases would have been prevented by if in the absence of cannabis use disorders in the younger males. So it is quite relevant. At last, cannabis has been long seen as a gateway, a transition drugs, and this is supported by several twin studies, which are the advantage of control for environmental and genetic influences in the clinical population, thus isolating the effects of the factor, so cannabis use in this case. But also a recent large meta-analysis show that the transition to move from THC use to opioid use is almost 3 times higher in the cannabis users in comparison with non-cannabis users. So this is a fact which can be easily explained by the alterations in the dopamine responses in brain areas associated with reward and motivation. This evidence emerged also by neuroimaging studies, which showed blunted dopamine responses in marijuana abusers in the ventral striatum, so decreased sensitivity to reward, which increased the vulnerability to substances of abuse, including opioids and other drugs, so poly abusers. However, recalling the role of the endocannabinoid system in the neurodevelopment, if adolescents, if the exposure to cannabis during adolescence can persistently affect brain and behavior, neurotransmissions and behavior, a profound impact could also be expected for in-utero perturbations of the endocannabinoid system when the CB1 receptor is already expressed in the fetal brain and orchestrates these key neurodevelopmental stages for a proper neurodevelopment. Among the things that may surprise you, or maybe not, is that the cannabis is the illicit drug most abused by pregnant women, with an increase in the reported use over the last decade, a steady increase actually in the last decades, in contrast with reports of decrease in the use of alcohol and nicotine, for instance. This is due also to the fact that pregnant women see cannabis as a harmless self-medication for many pregnancy-associated conditions, including nausea, but also anxiety, insomnia, and other conditions. But THC is able to cross readily the placenta and reaches the fetus and the fetal CB1 receptors in the brain, which are already expressed. Thus, it can, so THC and cannabis preparations can exert both direct and indirect effects, which pose a relevant risk for neurodevelopment. Early longitudinal studies, so in the clinical population, highlighted no association with severe pregnancy, detrimental effects on the pregnancy outcomes. However, upon a closer inspections in the pre-adolescent brain, in the pre-adolescent offspring, the emotional, cognitive, and behavioral alterations were observed, and they were consistent, were also associated with alterations in the expression of dopamine markers and opioid markers in relevant brain regions, such as mesolimbic brain regions. This is not easy to analyze because of the multiple variables included in the clinical studies, but in the animal model, we have the opportunity, the unique opportunity to study the causal effect of the THC exposure during pregnancy, and indeed the prenatal exposure to cannabinoids was able to, the early studies showed that this was able to alter the behavioral reactivity, cognitive functions, and also inhibit social play and social interaction in the pre-adolescent and post-adolescent offspring exposed to cannabinoids in utero. In our lab, we use experimental model, a rat model of gestational THC exposure, which mimics the gestational exposure to one mild cigarettes of cannabis, so 5% of THC along pregnancy. One of the first concerning outcomes that we observed was the increased vulnerability of the adolescent offspring exposed in utero to THC, according to our model, to alcohol drinking in experimental model of binge drinking, both in the male and the female offspring exposed during adolescence. But what can be behind, what's at the basis of this abnormal behavior for alcohol? We focused our attention on one of the most relevant modulators for alcohol-related behaviors, but also for the stress response, the neuropeptide Y. When we measured neuropeptide Y positive neurons in the mesolimbic brain regions of THC-exposed offspring, we measured actually a significant decrease in the medial prefrontal cortex, in the nucleus accumbens, and in the amygdala. I would like to say that MPY, this anti-alcohol neuropeptide, has an inhibitory effect on the synapse, both on the pre and the postsynaptic sides of the neurotransmissions. Thus, this suggested as a decrease, a deficit in the inhibitory tone, in the inhibitory modulation of neurotransmissions. That was confirmed by other data from our lab. According to this data, in parallel with a decrease in neuropeptide Y, we could measure an increase in the marker of the excitatory neurotransmission, so HOMER1. Thus, this confirmed as an imbalance in the inhibitory and excitatory transmissions, so hyperexcitability in multiple brain regions, including prefrontal cortex, nucleus accumbens, and amygdala. Actually, the increased vulnerability of prenatal THC exposed offspring during adolescence to alcohol was confirmed in another paradigm, an operant conditioning paradigm, where we measured an increase in the motivation to obtain alcohol, an increase in the relapse-like behavior after the deprivation in the exposed offspring, THC-exposed offspring, and compulsive-like behavior for alcohol in a task where the delivery of alcohol is associated to a punishment. So compulsive-like behavior in this kind of task. One of the most striking data that we could observe during this study was that just only half of the offspring exposed in utero to THC was able to learn how to gain alcohol in the operant paradigm, while all the offspring from the control groups, so non-exposed offspring, learned how to gain alcohol by pressing a lever in the operant chamber over 5 days. And this suggested us a deficit in learning and cognitive abilities. And that was actually in agreement, consistent with the recent evidence of a wide impact of prenatal cannabis on the hippocampal connectivity, so the key structure, the key brain regions for learning and memory. Thus, we wanted to investigate deeper this kind of effect of prenatal in-utero THC exposure on hippocampal dependent memory and hippocampal neurotransmissions. And we tested rats during adolescence, so in our model of prenatal THC exposure for hippocampal dependent spatial memory in the behavior, but also we measured the relevant markers of the excitatory neurotransmission and neuroplasticity, actually including endocannabinoid markers of which controls the excitatory transmission as well, the synaptic strength. In our model, actually, the mild gestational THC exposure did not induce overt memory impairment, because no difference was observed in neutral declarative memory in a simple recognition memory task, but a memory deficit was revealed when an emotional memory was evaluated. So a declarative memory for an aversive experience. This kind of evidence was also confirmed by other labs where when prenatal THC exposure was reported to decrease memory when the task requires an integration of multiple pieces of information, such as about location and the object to be remembered. So more complex cognitive task. And indeed, our most recent data on the evaluation of hippocampal dependent consequences of mild THC exposure in the adolescent offspring show that these offspring had an impairment in reinforce and aversion motivated spatial memory associated with an abnormal expression of the excitatory and endocannabinoid related markers of excitatory neuroplasticity in the hippocampus, suggestive of hyperexcitability. Thus, overall, our data contributes to characterize the phenotypic consequences of gestational THC exposure. But the hyperexcitability emerged as a possible rescue, possible target for rescue strategies. And in this regard, cannabidiol, the second most abundant phytocannabinoid in cannabis, as a treatment indication, therapeutic indication in brain conditions characterized by hyperexcitability, including epilepsy, treatment-resistant epilepsy in the pediatric patients. In addition, cannabidiol was reported to enhance memory and cognition in a very young population. So taking advantage of the peculiar cannabidiol pharmacology, which is very different from THC's one, and is able to modulate, to improve inhibitory, which is able to improve inhibitory neurotransmissions with a large, a wide spectrum of actions, was actually our try, was tried in our model to modulate the neuroplasticity during adolescence. Indeed, during this kind of developmental moment, as you already So before the endocannabinoid system goes by a multiple dynamic change. Indeed, CB1 receptor expression is kind of similar to early gestation, while the expression of one of the most relevant metabolizing enzyme for endocannabinoids is very high. And this can be blocked also by the repeated administration of cannabidiol, so pharmacologically blocked by cannabidiol administration during adolescence. In our experimental model of prenatal THC exposure, cannabidiol administration, repeated cannabidiol administration, exerted a positive impact on the cognitive scores, both in reinforcement motivated spatial learning and aversive motivated spatial learning, and was also able to dampen the expression, the abnormal expression of endocannabinoid related markers and excitatory related markers of neuroplasticity in the hippocampus. So in controlling actually the hyperexcitability in the brain of THC exposed offspring. Consistently, agents pharmacological agents able to improve inhibitory neurotransmissions can also, thus curbing hyperexcitability in the brain, such as the neurosteroid pregnenolone, proved effective also in rescuing the synaptic and neurological dysfunctions in the prenatal THC exposed offspring in a similar model of THC gestational exposure. Thus, in our current activity, the exploration of alternative and safe strategies to modulate the developing endocannabinoid system is ongoing, and this research is including the testing the polyunsaturated fatty acid supplementation during gestation and lactation when the exposure to THC can occur. Overall, our data is suggestive so far suggestive of enduring the consequences on the emotional and the cognitive function and alcohol addictive like behavior in male rat offspring. Thus, these behavioral alterations can result from the dysregulation of the inhibitory excitatory signaling in mesocortical limbic system and the increasing hyperexcitability in the brain. However, if the role of science is to the definition, the identification of early in the phenotype and molecular targets to the evaluation able to be druggable, for the evaluation of safe rescue strategies, the education is key for spreading information, knowledge on the role of the endocannabinoid system in all the stages of neurodevelopment. So, and the prenatal side, also the adolescent side. That would also inform and guide the political interventions in order to protect the vulnerable population and thus to have a very relevant, efficacious action. However, a science-based communication strategy can nowadays make a difference. You know that influencers and role models have a wide impact on a general population, and in this regard, you may have heard about Rihanna's recent interview where she states that she is actually abstaining from using cannabis during pregnancy. But good things is that we all can use the internet and the social media to achieve a large-scale communication, and each of us can contribute to spread evidence-based information, science-based communication to protect the developing brain. So I would like to say thank you, because this is my last slide, so I would like to say thank you for following. Thank you, ISSUP, for this opportunity. And Professor Cannizzaro and her lab we are all at your disposal for further information and further curiosities. So start like, okay, maybe.
Ok.
Ok.
Grazie. Grazie davvero alle...
Thank you so much. Thank you so much, Anna. So thank you to Professor Cannizzaro, and thank you, Anna. There are 4 questions which Peter can read out.
Both Professor Cannizzaro and Dr. Brancato for their very interesting work on the effects of cannabis on the brain. There are lots of questions. I just want to concentrate on a few of these questions. The first question is very interesting. It reads like this: based on the detrimental consequences of early exposure to cannabis use across the lifespan, at what stage of human development should prevention of cannabis use be implemented? When do we begin? What stage of a child's life, of an adult's life, do we begin with prevention? I don't know whether Dr. Brancato or Professor Cannizzaro.
I can answer that because I've been talking about the neurodevelopment of the brain. So if we consider that the brain is still maturing after adolescence, which means after 19 years old, I mean, this means that actually until the full maturation of the brain, it is not advisable to take any cannabinoids containing products. And I am not talking just about cannabis, but also the so-called synthetic cannabinoids, which are analogs of the phytocannabinoids, but also they possess a stronger pharmacological activity. It is interesting, the question, because I can read it on the other way, which means when can I get to the good properties of cannabis? So I don't want to go into this, but actually we should all remember that cannabis and THC are able to produce modifications in the brain neurotransmitters, and so the thing is, even if cannabis is natural, we have to understand that cannabis and cannabinoids produce pharmacological effects in the brain.
Okay. Another interesting question is, if cannabis is so dangerous, its effects are so dangerous on the brain, why is there so much pressure at the social and political level to legalize it? I don't know whether this is a question, Dr. Brancato or Professor Cannizzaro. Why is there so much pressure in society today to legalize cannabis? If it's so dangerous, if there's so much evidence from the medical point of view that it is dangerous, especially growing up in children?
Okay, so I'll take this again, and then I'll stop talking. I'll leave Anna the other questions. But the thing is, I think they put the problem, the issue, on another field, which is not really safety, which is not really the activity on the central nervous system, on brain development. Usually when you go to these debates about it's good or bad to legalize, we usually hear, and it's correct, I can agree, that legalizing cannabis means to avoid the black market of drug selling, and this is true. But on the other side, we cannot hide ourselves behind the finger. The problem is real. So if we believe that legalization is correct for adult people, we have to be very strict, trying at least to be very strict in stop young people, help young people, protect young people from consuming cannabis in vulnerable ages, in vulnerable periods of their life. So I think it's in between political, social, and yes, issues, but the safety question, the safety profile of cannabis is not really well known, and sometimes I think is underestimated, even in debates on the web or on TV.
Okay. Last now, Dottoressa Brancato question, which was, I think, was the first question. The second question. No, the third question. Is marijuana a gateway to the use of other drugs? You answered this in a way. Could you develop that again from the studies you mentioned?
Yeah, the most recent evidence is actually a large meta-analysis, which included hundreds of people, showing that cannabis users are more prone to use to go into opioid use and develop a more problematic opioid use pattern of consumption. Indeed, this can be related to the age of onset of cannabis use, to the dosage, and so the heavy cannabis, heavy THC exposure during developmental periods, because of the many modifications in CB1 receptor density in many brain regions, controlling reward sensitivity and prefrontal cortex also, so inhibitory top-down control. These modifications actually make the brain more vulnerable to other drugs of abuse. Opioids is the most, maybe one of the most dangerous, but also alcohol and other drugs. So poly abusers, the poly abuse behavior associated with cannabis use and abuse also is a fact.
There's another question just arrived. Do lower concentrations of THC produce less harmful effects on the developing brain, or do synergistic effects exist? Exist from other compounds, terpenes, flavonoids, etc. So can I repeat it? You want me to repeat it?
I think I can also read that. A synergistic effect actually can be detected, but THC and cannabidiol in the in the preparation of cannabis have different profiles. So cannabidiol actually counteracts THC, many of THC detrimental effects, the thing is, we cannot say how much THC is, which is the concentration THC in cannabis preparation, which are not controlled, because cannabis is sold, black market. We cannot say how much THC. So it's not. There's no, a minimum concentration allowed, I believe, in the developmental time window.
Okay, there are lots of questions arriving now. I just want to choose some of the questions. Like, I think we have time for a few more. This is an interesting question. What are the withdrawal symptoms of cannabis? I don't know who's going to answer that.
It's many
symptoms?
Symptoms include the cannabis craving, but also difficulties in sleeping, anxiety, anorexia, nausea, many symptoms which are related to the decreased availability of CB1 receptors, actually. So,
and this is another question connected, I think. Sorry?
Would like
this
to
is another
say
question
also.
connected.
Okay.
Is there a specific timeline to say that marijuana use is already chronic? When is it chronic? When does it become chronic, the use of marijuana? It's connected.
This is interesting question.
Can I say a word on that? Because actually we should consider what we really mean by chronic. Because the thing is, chronic THC, chronic cannabis, takes to dependence. So the dependence to be dependent on cannabis can be a condition. You arrive differently according to the organism, according to every individual organism. So yes, definitely. So chronic means that you take the substance regularly. That you have also to remember that THC is metabolized and leaves the organism very, very slowly. So you can have THC in your blood for different days, as Dr. Brancato has already explained. So chronic means several consumption, several administrations of cannabis. When you take cannabis chronically, you feel this feeling of, desire to take cannabis again. And this is the basis of dependence, of course. So as far as you can stop, as far as you can control it, but really control it. So it means after weeks, you don't feel the craving for cannabis, then that's not dependence.
Okay. I think do we have another time for another question, Joanna?
Un'altra domanda e poi io volevo chiudere, diciamo.
Okay. Another question. The last question. Because there are lots of questions arriving. I'm just going to read this. Could long use of cannabis create mental illness? And I think you've answered that during your speeches. But can this be used in a criminal court as a defense in a non-violent case? This is the question. What about the legal consequences? Can the mental sort of issues be used to justify in a criminal court sentences? I think this is a very legal, it's a legal matter.
Well, the legal side, I'm not an expert of the legal side, but cannabis is a risk factor. Many conditions see environmental risk factor, which you can control you can avoid to control the occurrence, the possibility to develop a condition. So cannabis is one modifiable risk factor for psychosis, but is not the only one.
Obviously, it's connected to alcohol consumption, same sort of idea. how does alcohol...
Environment, so many experience in the life, in a vulnerable population, in a vulnerable individual.
And Peter, I would like to add that maybe it is also important to make into correlation the time of the case and the presence of the substance, because, you know, when you have people using drugs, they are caught for, you know, crimes or whatever, you have to be able to connect the precise time of the crime with the presence of the drug in the organism. This is not so easy, okay?
Okay. It's been interesting questions, interesting answers. Joanna, do you want to conclude?
Sì, allora, intanto...
I can see the debate was quite lively, and these are the last minutes of our webinar. I'd like to thank Carla and Anna for these very important presentations, which have highlighted the importance and care from the beginning of the use of these substances and neurodevelopmental stages. They made a reference to new elements, the importance of the study of epigenetics and the consequences this has on the development of embryos during the regulation of all the metabolic patterns. I also would like to say that genetic studies focus on pathological dependencies or pathological addictions, and the topic is not related to momentous transmission. Many parents or families ask us, is pathological addiction hereditary? Maybe this is the topic for another webinar. Carla suggested, made a reference to epigenetic and action mechanisms. It's also important not to give to genetic that determinism in the framework of pathological addiction, because we should work on empowerment, on prevention, development of healthy lifestyles, mirroring the fact that the environment has an impact on gene expression, but which are also closely related to how the individual manages to develop the skills for self-protection. The data introduced by Anna are impressive. The use, I was surprised to see the increase of the use of substances during pregnancy, but hopefully the fact that we can prevent and raise awareness, we can teach women on how dangerous it is to use these substances, and at the same time we should have the possibility to find a cure to avoid harmful effects on the offspring. Thank you for your contribution. Thank you, Peter, Anna, and Carla, and Carmelo, the interpreter, who have enabled us to have this webinar in an effective way with 470 participants. We're very happy with that. Thank you. Thank you. I hope to see you soon and carry on with our training and education.
Grazie. Grazie.
Olivia, I don't know if you can hear me.